So the difference between mitosis and meiosis is clear. And we've made it clear over the last 3 weeks with some students. One student specifically asked for us to kind of hit it one more time. So I'm going to go ahead and hit it and send it to you before the quiz. And we're looking at meiosis, but before we go on to meiosis, let's remember what mitosis was done for. It was, number one, making exact copies of all 23 pairs. The reason you would make an exact copy, number one, I should have put this as A, is to grow, to go from that one cell to that trillion cell organism that is who you are. Next, we have to heal in case we get wounds or something happens. Of course, replacing cells that have died. Now, these two are similar in that they both result from the death of a cell, but they're different in the reasons why the death occurred, right? So be careful in looking at that and don't get confused. Of course, another reason would be to asexually reproduce, but we don't as humans do this, but plants do it and other organisms do it, and they have ways of increasing genetic variation, but that genetic variation isn't as robust as you find when you do meiosis. So creating these cells and remembering why we want to create these cells, we have to think about the fact that we need cells that are not the same. We don't want to be clones of our parents or like somehow divided in half of having a both large and small nose, or having exact half of our DNA being a clone from our mother and a half exactly our DNA being cloned from our father. We don't want that. We want to recombine the DNA. We have to have some way of introducing genetic variability. All asexually reproducing organisms find a way to do that. Humans do sexual division, and that's true of a lot of eukaryotes, I should say. And the key to doing that is remembering that when we have two cells and they have that 2n version of the number of chromosomes, so that's in humans, that's 46, right? And another 46 over here, or 23 pairs, right? So that's 23 pairs. Remembering that we're dealing with 23 pairs of chromosomes, so 1 through 23. When they combine, if we just took that and combined these two into a single cell to make a child. So if we want to make a child, we want to take something from mom and something from dad, and we want to go ahead and combine them and make this new person. We don't want to make 92 chromosomes. We want to keep that number at 46. So you can think of this in two ways. Think of it in mathematically. How is it that I take 46 chromosomes, 23 pairs? And I want to remind you of this. This is something we hit a lot in mitosis, but I want to go ahead and back and I want you to think about it. 23 pairs means that I have this chromosome 1 I'm going to have one from dad, and I'm going to have another one from mom. They're going to be exact copies of one another, okay? So dad's is going to look like this. It's going to be, when I say exact copies, I mean, I shouldn't say exact copies. They're not going to be exact copies, but they're going to be homologs. And what that means is they're going to have the same genes on them. So mom's over here is going to have a gene A, right? It's going to have the gene for ear color, or ear size, attachment or not. And dad is going to have the same gene in the same place over here. Dad might have attached earlobes. I mean, that's a big A. And mom might have unattached earlobes, and that's a small a. So that's true for all the genes on both these chromosomes. They're all going to be the same in the sense that they're all going to code for the same characteristic. But each parent is going to give you a slightly different version of that gene that's going to make a difference. It's going to make some difference. Whether that difference is important or not, well, it's another discussion. So you have you have this allele. We call it an allele. And these alleles are just different versions of the gene that are found on this chromosome. They're both chromosome number 1. So that's where we get that pairs. That's where we get the pairs. And that's through chromosome 1 all the way through the 2 and 3, all the way down to 23. All 23 pairs. This is happening. Okay, so we can't really combine two cells and combine them into one and get 92 chromosomes, because that's gonna cause cellular death. It's going to cause issues. Anytime you have more than two copies of the gene, it's an issue. Our bodies and most vertebrate animals have only two copies of the genes of every gene that they have. Now, there are exceptions in plants. For instance, corn has an incredible number of genes, but copies of genes in any particular tissue. But humans specifically, and mammals in general, vertebrates, and probably most living animals, and what we're looking at is combining 46 of each going to 92 means death. And we can't have it. So we don't want, that's not what we can do. We can't accomplish this successfully. So we have to find a way to combine two cells that have 46 chromosomes and get a new person. This is a new person, right? This new person has to have also 23 pairs, or 46. How do we go from combining two 46s and get 46? Because this one, getting 92, is wrong. We're going to end up with death, right? And we don't want that. We want the new person. We want this new person to survive and be a newborn baby girl or baby boy and be happy with it. So how do we get it? There's mechanics. If you think about it mathematically, there's only one way to do it, right? Well, maybe there's more, but there's one obvious way to do it. And that's that you have this one and you have this one, and you combine those together. So you would have, if you want to combine, you got to have one that has only 23, one copy of each of the 23s rather than two copies of their 23. And another cell that has one copy of the 23 to get, and remember, because that's possible, because this pair would be chromosome number 1, for instance. There's two chromosome number 1s. They have the genes for each of the characteristics that are found on that chromosome. So it's not like if you don't get mom's chromosome for some reason, you're not going to be able to have eyeballs or brain. You're going to have those characteristics because you have a copy from dad that has that gene on it. And the mother of your mother, who's going to give you that other chromosome number 1, also has those same genes on that chromosome. The difference is you're going to have these different alleles. So these are going to be different, right? But the genes are going to be there. So that's an important thing to think about. Okay, so how do we accomplish that? Well, in meiosis, what we're going to do is, and this is something the book talks about, and we'll go ahead and just say it, even though it's a really broad generalization, it's really simplistic. We'll say meiosis, really, when we're looking at meiosis, we're looking at doing mitosis twice, right? And when we're going through mitosis, the first step of mitosis is prophase, and we do prophase in meiosis, but to make sure that we designate the differences between prophases, we call this prophase 1. Now in prophase 1, the big difference between that and mitosis, and why this is so general, it's really, I should put in quotation marks, because it's really just too general to say it that way. Because in prophase 1 of meiosis, you don't really, there's some similarities in that the chromatin does condense into chromosomes, and they are paired up, right? Just like, because they've condensed, you have that kind of X-looking shape, and the reason you have that is the sister chromatids that are identical. The one from mom right here, and the other one from dad that you got. You're going to try to make a sex cell. You're going to try to make a cell that you can combine with your wife's in the future, that allows you to create a new being, a new human that's healthy. And so in order to do that, what you need to do is you're going to need to have this DNA. And then prophase 1, the way that the cell first gets them, is the way it's going to be different from mitosis, is that these two chromosomes that normally, right, in mitosis would line up along the mitotic plate in a straight line, these chromosomes are going to come together in prophase 1. And they're going to form this thing we call a tetrad. So it's going to be kind of a little funky looking thing that looks like this, right? And there's one chromosome number 1, and it's going to be attached to the other chromosome number 1 right there in the center. And these chromosomes, the interesting thing is this is going to form a tetrad, or 4 chromatids. And wherever these tetrads have a chiasma, right, where the place in a region where they're going to be able to switch, they're going to switch their DNA. So this is where crossing over occurs. And this crossing over occurs in prophase 1. What it does is it increases variation. This is a really important difference between mitosis and meiosis. These tetrads actually allow for increasing genetic variation, because instead of having 4 chromatids, 2 exact copies, right? This chromatid has the same genetic information as this, exactly the same. Where this chromatid over here has exactly the same information as this chromatid. They all have the same genes, but where this one, the one from mom here, might have little a, the one from dad would have big A. Both of these are exactly the same. But by crossing over in these regions, here, let's say it crosses over here and here, by crossing over in these regions, what you create is this section of DNA that is now, instead of having 4 chromatids, 2 from each of the parents, right? You have 4 different chromatids, where before you had 2. These 2 would be the same, and these 2 would be the same, and these 2 would be the same. Being chromatid 1, 2, 3, and 4. All 4 now are different after crossing over. After you cross over, they're all 4 different. And that, I think, it's obvious by its nature is going to make the in the end, you'll see that you're going to have 4 different possibilities. You've increased variation. You no longer have big A, and little a. You have a combination of that, and that's important. So, okay, after prophase, after they've done their crossing over, they're going to line up the metaphase plate. Now, remember, we're talking about one cell at this point. Here's your middle line, right? Their metaphase. But instead of having those chromosomes where you have, like in mitosis over here, let's say this is mitosis over here. In mitosis, they would line up, and you'd have one chromosome, one chromatid from one chromosome with one chromatid from dad, right? Chromosome 1 copies. This dad chromosome over here, and this dad chromosome over here. These chromatids would move in opposite directions, right? And you'd have another chromosome number 1 lined up exactly the same way. And then these chromosome number 1s each would move into the new cell area, the new nucleus area. That way you would get chromosome number 1 from mom and 1 from dad, and they would go in each of these new cells. It's easy to understand. So you have in mitosis, you're going to get this sameness, right? And when they're divided, you have 2 cells that are exactly the same in the number of DNA. We don't want that meiosis. We want 4 cells that are different, and they're going to need to be half the DNA. So the way the cell does is it takes this newly recombined DNA. So you got this chromatid here. That's exactly the way it was originally from dad. And then you have another chromatid that's still attached to it. But this chromatid has something different. It has a little section of mom's DNA. And it has up here a section of dad's DNA. And this is attached, right? This is attached to a chromosome from mom, which has some of dad's DNA in that same area. So they switch that little bit of DNA, and all the rest of it is the same. And when you're looking at it, these, oh, sorry, that should be a different color. So these here, this pair, this homolog, remember we call these homologs. these homologs here are both going to move in this direction, and these homologs here are going to move in this direction. Now, they're both chromosome number 1 or 2 or 3 or all the way through 23. But the difference between mitosis and meiosis is that over here, chromosome, you're going to get 23, and the end of mitosis, you're going to get 23 pairs or 46 chromosomes over here. And you're going to get 46 chromosomes over here. And you're in both of these copies, you're going to have half from mom and half from dad in both of these. Half from mom and half from dad. And they're going to be exactly the same genetically. You couldn't tell the difference between these two cells genetically. But here, that's not the objective. Here, we're going to move in the first division, these two here, these two homologs. Again, homologs means they're the same, they carry the same genes, not the same alleles. So these two homologs are going to move in the direction of making a new cell on this side to the right, I mean to the left. And these two are going to go make a new cell to the right. Again, this happens 23 times. So in the end, you're going to end up with two cells, one having a chromosome that has a chromatid that's 100% dad's, right? And you have another chromatid attached to it. They generally form that kind of X pattern to it, an X shape, that has a piece of mom's DNA. Now, in all the illustrations that you'll ever see on this, they do their best to make these color combinations for you. And the other cell will be exactly the opposite, right? It's going to be mom's set. It's going to be mom's chromosome. The two chromatids. That's why we make that distinction between chromosome and chromatid, so we can have this discussion. Otherwise, we go crazy trying to describe the difference. So this chromosome having one chromatid, 100% moms, and the other one in this example having mostly moms and some of dads. Well, where to get that dads? It got it from that crossing over that happened that we discussed a minute ago. So these two are essentially already different. They've already half the DNA, half the number of chromosomes, right? Already. But we still have two copies in each cell. We don't want that. We want one copy of each chromosome in each cell. So how would we do that? Now, this is obviously after anaphase 1, after telekinesis 1, and after the cell division. We have this, after cytokinesis, we have these two cells. This is all done with 1. Now we're going to go into meiosis 2. We need to get this divided again, but this time we're not going to duplicate the DNA. The reason we got these two copies to begin with was because we duplicated the DNA. The reason that the crossing over happened in prophase 1 is because you had all 4 of the chromatids together in that tetrad. Well, now you have 2 chromosomes. One chromosome, 2 chromatids in each cell. Now each of these are going to divide. And they're going to go through all the steps of mitosis again. They're going to go through prophase 2. This time in prophase 2, there's not going to be any crossing over. They're not going to line up like they did. They're going to line up in metaphase. They're going to line up just like they did in mitosis as chromosomes, but you're only going to have one copy of each chromosome, and they're going to split the chromatids so that you get one chromatid for each chromosome. So this is now a chromosome. Remember when we talked about it, when chromatids split, we call them chromosomes. So you have one chromosome here, chromosome number 1 from dad. Then you have one chromosome that is a kind of a combination of mom's and dad's DNA, right? That recombined DNA that happened at crossing over. You have this chromosome that's partly mom and partly dad's. And you can see it here. This is partly mom and partly dad's, and it came over here. It's separated, independently segregated. And this one independently segregated over here. Now that's key, that they're independently segregated, so we can't tell where they're going to end up, okay? That increases variation. Now, over here in this other set of cells, you have, again, a chromosome that's 100% mom's. And then you get a chromosome that's mostly moms, and that has some of dad's DNA at the tip here, which is what you see here when we look at this. This is what crossed over. Remember, it crossed over and took this piece, and this piece came over here. And you can see that these are kind of mirror images of each other. And now you have 4 cells with half the DNA, not just half the chromosomes, because up here you had half the chromosomes, but you still had the double DNA, right? Now you only have 1 copy of each, a single chromosome, 23 chromosomes, a single copy of each chromosome in each of these 4. I'm only drawing 1 chromosome, but there are 22 others. And one of them has been recombined. There's actually 2 of them have been recombined. So now you have 4 different individuals that can come from these 4 different, what we call again, gamete cells. Gamete cells, right? So these gamete cells are are half the cells, so we call them haploids. Again, these are haploid cells. They only occur in the testicle or the ovary. They're only for making a new human being. So meiosis only happens to make new human beings. You only have the DNA, make these gametes or make these haploid cells. The only time this happens is to make this child that we're trying to make that's going to have a healthy combination of DNA. All right, so that's meiosis, and let me summarize it by looking at this diagram, which again you'll find in your PowerPoint. And this diagram shows you clearly when we're looking at a 2n, right? In this case, 2n equals 4. So this organism is not a human, right? So 2n is 4. In humans, 2n would equal 46 or 46, right? And so 2, because n is the number of chromosomes you have. You have 23 chromosomes, but 2 is because you have 2 copies of each chromosome, one from mother and one from father. That's in any sexually dividing organism, sexually replicating organism. And so before you divide in mitosis, if we look at just mitosis, what you're doing is you duplicate the DNA. So you have 2 copies of each, and that's why you got those X-looking things, because each strand, each chromatid is 1 copy of the chromosome. So here's 1 copy of dad's, and here's another copy of dad's, right? And then here's 1 copy of mom's, and here's another copy of mom's, whatever this beast is. Because it's a 2n of 4, it's not human. Over here in prophase 1, you have meiosis. You're doing the same thing. Notice that this, prophase 1, in the beginning of prophase, this and this are the same. Now, what happens here that does not happen over here is that we get crossing over. These 4, see how they lined up? The difference is that the way they lined up, when you look at it, this particular set are lined up in a tetrad. And that tetrad is important because it allows for crossing over. You'll notice that in the other end of that, you also have a tetrad. So you have 2 tetrads here, one set here and one, let me use a different color so you can see it clearly, okay? You have 2 tetrads here, and one of them is, the chromosomes are larger, the other ones, the chromosomes are smaller, but when those tetrads get together, they switch that DNA. You can notice it here. You'll see the little changes that occurred. There's a little green in the tan one, and there's a little tan in the green one, just like I showed you a while back. So it's because of that, these tetrads, when they align, when they first do here, we have crossing over. All right, so this crossing over is important because it allows for, increases that genetic variation. In the other one, prophase, in prophase and metaphase of mitosis, they line up straight up and down. So these 2 pairs of these homologs, we call these homologs, they don't come together into a tetrad in mitosis. They just line up and then they divide. Well, you get 1 copy in chromosome 1 here from mom, 1 copy from dad, 1 copy chromosome 2 from dad here, 1 copy chromosome mom here. They line up, and these homologs split by 1 chromatid over here, 1 chromatid over here, giving you 2 copies, right, of each chromosome. So you end up with a 2n copy over here. So you have 4 over here and 4 over here. So in mitosis, you end up with 2 exact copies of the cells. In meiosis, that crossing over mixes up some genes, so already you can't have exact copies of the cell. But then when you divide when they divide the pairs. You see the 2 chromatids stuck together. So already now you're drastically different, because here you have moms with a little bit of dads. Here you have dads with a little bit of moms. So there's no way that these 2 are going to be anything alike. And then you split them up again, and you get one radically different with a recombined chromosome and a regular chromosome, and a recombined chromosome and a regular chromosome throughout all 4 copies. Now, all 4 of these are going to become 4 individuals. And when you combine these 4 eggs or sperm with 4 different eggs or sperm from with another individual from egg or sperm from another parent, you're going to get a whole new being. This can be much different than the parent. This is very much unlike asexual division that happens in things like bacteria.